Today, we are attempting a true deep dive into what might be the single biggest engineering challenge humanity faces this century: building a permanent, safe, and, well, sustainable home on Mars. And this isn't just about sending astronauts. It's about establishing a lasting presence on a world that is fundamentally hostile to us.
It really is. The conditions are, frankly, horrifying.
Horrifying is the right word. We're talking about a planet with an atmosphere so thin the surface pressure is, what, less than 1% of Earth's?
Less than 1%, and
Yeah.
it's 95% CO2, constantly bombarded by cosmic radiation. There's no protective magnetosphere to speak of.
And the temperature swings
Yeah.
from a bitter minus 90 degrees Celsius to, on a good summer day, maybe 26 degrees.
Maybe. And that's exactly why you can't just rely on Earth materials.
Right.
The cost of launching basic supplies, steel, concrete, it's just astronomically expensive. Logistically impossible, really. To build a home there, we have to use what's there. In-situ resource utilization, or ISRU.
We have to transform the Martian soil itself into building material.
We have to.
And that's our mission today. We're focusing on this remarkable piece of research led by Dr. Shiva Krishnath. It was published in Frontiers in Microbiology, and it proposes we bypass traditional engineering entirely.
Right. No high heat furnaces, no complex chemistry.
No. Instead, they want to use Earth's toughest, tiniest organisms, microbes, to convert loose Martian regolith into protective structures. It's literal biological engineering.
Where life itself becomes the construction tool.
Exactly.
And what's so fascinating here is how multi-layered the objective is. This isn't just a civil engineering problem.
Yeah.
It's a systems engineering problem. They're integrating materials, science, biology, architecture, everything. They're trying to create not just a shelter, but a complete protective and potentially regenerative life support system.
And the timing on this just feels so perfect. We have NASA's Perseverance rover right now collecting samples in Jezero Crater, and we know there are carbonates there, minerals that usually form in water. That suggests ancient, maybe even habitable environments.
And if you connect those dots, it raises a really profound question. If biological processes, likely microbial ones, helped form minerals and rocks on ancient Mars, could we use similar modern strategies to build our future?
So the history of Mars could actually provide the blueprint for how we survive there.
It's a beautiful idea. It suggests the planet itself might hold the key.
Okay, let's look at the foundational science here, this idea of biomineralization. Most people think of life as organic, soft. But this is the process where microorganisms, bacteria, algae, produce hard minerals as part of their metabolism.
Yeah, these organisms are truly nature's silent engineers. They've been transforming Earth for billions of years. They gave us our oxygen atmosphere, but they also built enormous structures. Think of coral reefs,
Right, or ancient stromatolites.
Yeah.
They prove this can be done on a massive, resilient scale.
And the specific pathway Krishnath's team is focused on is called biocementation.
This is key. Traditional cement manufacturing is incredibly energy intensive. You have to heat limestone to blistering temperatures.
Biocementation just flips that on its head. It uses microbes to generate natural cement-like materials, calcium carbonate at room temperature.
Which is absolutely critical for a low-power environment like Mars.
And this research draws its inspiration directly from extremophiles organisms that thrive in the harshest places on Earth.
So like acidic volcanic soils, deep caves.
Exactly. High pressure, high salinity. If they can survive those conditions, they've got the baseline resilience needed for Mars.
But that leads to a huge question of safety, which the researchers address head on. If you're introducing life to another planet, you have to be absolutely sure you're not causing interplanetary contamination.
That is the paramount safety concern. The whole idea is to find microbial pathways that are contained, that build strong materials, but pose zero risk of becoming invasive. You have to select strains that are well understood and can be carefully controlled.
Which makes the choice of organisms absolutely vital. So let's meet the construction crew. The research centers on this powerful partnership, what they call the dynamic duo.
And this is where the biological complexity just shines.
Yeah.
You have two very different, highly specialized bacteria that work together.
First up, the primary builder, Sporosarcina pasteurii. This is the one that does the actual cementation.
It's famous in materials science. It produces calcium carbonate through a process called ureolysis. It's the cement factory.
The cement factory. And then second, we have the protector, Chroococcidiopsis.
A very resilient cyanobacterium. It's basically a single-celled photosynthetic organism.
And it's already been proven to survive in incredibly harsh simulated Martian conditions in labs.
Yes, and the synergy between these two is the key to the whole thing. They have a strict division of labor. See, S. pasteurii needs a somewhat buffered, less toxic environment to do its job efficiently.
Okay, but that brings up a major chemical challenge here. The process, uriolysis, it needs urea to work, and urea requires nitrogen. Mars is notoriously nitrogen poor compared to Earth, so where do they get the feedstock to sustain this?
That's a sharp insight, and it's a huge research area. I mean, you could theoretically synthesize it from atmospheric nitrogen, but that's very energy demanding. The more elegant solution, the one the researchers favor, is a closed-loop system.
Closed loop.
The nitrogen, the urea, would be recycled from astronaut metabolic waste.
So human waste, which is a huge logistical problem in space, becomes a valuable resource for construction.
Exactly. Waste becomes the nutrient for the cement factory. It's incredibly efficient.
Wow. Okay, so back to Chroococcidiopsis, the cyanobacterium. How does it protect the cement boulder?
Its role is multifaceted. First, because it's photosynthetic, it releases trace amounts of oxygen. It's not a lot, but it helps create a slightly more habitable microenvironment for S. pasteurii in the regolith.
But its main job is protection, right?
Right. The primary protection comes from its secretions. It secretes something called an extracellular polymeric substance, or EPS. Think of it not as simple sludge, but as a kind of complex biological hydrogel. It's a sticky polymer that forms a matrix around the cells.
And this EPS acts as a shield.
A fantastic UV radiation shield. It absorbs or scatters those harmful ultraviolet rays on the Martian surface. It lets S. pasteurii survive long enough to do its job.
So the cyanobacterium provides the shield and a little bit of oxygen, creating a sort of micro-oasis. And in return, what does the cement factory, S. pasteurii, do for the structure?
Well, once it's protected, it gets to work. The uriolysis process causes calcium carbonate to precipitate, and that acts like a microscopic glue. It binds the loose regolith particles together, and the polymers it also secretes further strengthen that material matrix.
So you're turning loose soil into a solid concrete-like material.
Exactly. And the researchers envision this bacterial co-culture, mixed with Martian soil, as the perfect self-healing feedstock for 3D printing habitats.
That merging of robotics and biology is really what sets this apart. It's a living material.
Yeah.
But you mentioned the benefits go beyond just structural integrity.
Oh, absolutely. The structure is critical, but the life support benefits are just as immediate.
Let's focus on the most obvious one, oxygen. Chroococcidiopsis is photosynthetic. It's actively converting CO2 from the Martian atmosphere into oxygen. That O2 could be harvested and fed right into astronaut life support systems.
It could. It would reduce the need to rely solely on mechanical systems or stored oxygen. But then if you connect this to the long-term vision colonization terraforming, it gets even more profound.
Oh, so?
Remember the byproduct of uriolysis, the waste product.
Ammonia.
Right. Sporosarcina pasteurii produces ammonia.
Which is a key component for fertilizer.
Precisely. That ammonia could be the critical link for establishing closed-loop agriculture in the habitats. Future colonists could potentially use waste from the construction process to grow crops.
So you're introducing nitrogen cycling and oxygen production.
You are. And while full-scale terraforming is, a millennial-scale project, these are the tiny foundational biological steps that could eventually lead to that.
It's an incredible closed circle. Human waste feeds the builder. The builder provides the structure and fertilizer, and the protector provides the oxygen. It's a self-sustaining ecosystem built into the foundations.
That's the vision.
But, the first human habitat is slated for the 2040s. As elegant as this all sounds, the path to validation must be filled with some serious hurdles. What's the holdup?
The most immediate one is logistical, and it's all about materials. The recurring delays with the Mars sample return mission, it means researchers can't get their hands on actual Martian regolith. They're forced to rely on simulants.
Fake Martian dirt.
Fake Martian dirt, yeah. And that really constrains how accurately they can predict real-world performance.
So what are the key differences there? What makes the simulant so limiting?
There are a few major things. Real Martian regolith contains perchlorate salts, which are toxic and very reactive. Simulants also struggle to perfectly replicate the exact mineral phases, the particle size, and the unique electrostatic properties. And maybe most importantly, we don't know for sure how the microbes would interact with authentic Martian water content and chemistry.
So the models they're building are only as good as the fake dirt they're using. What are the key areas of active research needed to bridge that gap, assuming we don't get the samples anytime soon?
Well, the first area is still centered on astrobiology and materials science. They need to optimize the microbes to withstand the combination of stressors, not just UV, but the cold, the low pressure, all of it. They have to use really advanced predictive modeling with the best simulants they have to forecast performance.
And the second major area sounds like a whole different universe of problems: hardware and engineering.
It is. The major technical challenge for manufacturing is the robotics, the 3D printing process itself. You can't accurately test autonomous construction for Mars in Earth's gravity.
Right, Martian gravity is only about 38% of ours.
Yeah.
So you have to somehow simulate low gravity for giant construction equipment.
It's incredibly difficult. Testing requires specialized low-gravity analogs like complex balancing systems or parabolic flights, which are costly and brief. But the biggest challenge isn't just the environment, it's the control algorithms for the robots. You need protocols that optimize the 3D printing precision in that low-gravity, low-pressure world. You have to ensure the bioconcrete mix flows correctly, cures uniformly, and maintains integrity layer by layer, all without a human standing there to fix it.
That makes it sound less like mixing concrete and more like programming a self-assembling biological system to work perfectly in a vacuum, with the safety of humanity resting on, a few billion tiny bacteria.
That's a pretty good summary.
So to wrap this up for you, the listener, this research represents a radical paradigm shift. It integrates astrobiology, geochemistry, material science, robotic engineering, and it really changed the definition of building on the red planet. It proves that the smallest, most robust life forms we have might be the only viable way for us to make our biggest leap into space.
Which leaves us with a really powerful final thought. If the future of human architecture on Mars relies entirely on harnessing Earth's basic biological processes to turn alien soil into breathable concrete, what does that tell you about the fundamental, relentless power of biology? Life, once it's established, consistently proves itself to be its own first and most enduring engineer. So if we can solve planetary colonization this way, what other seemingly insurmountable challenges right here on Earth, from infrastructure repair to sequestering pollutants, could we redefine by learning to better harness our microscopic colleagues?